barrier works by forming the selective permeability, in which it means the restrictions of molecules entry and exit to the cell. Next, we have the glycolytic. Glycolytic is mainly used for cell recognition, and then we have the glycoprotein. Glycoprotein is involved for holding the correct cells together from tissues such as in muscle or nerve tissues. Next, we have the cholesterol. Cholesterol disturbs the phospholipid cross-stacking arrangement to keep them fluid, and that also increases the flexibility and stability of the phospholipid bilayer. So, cholesterol do a same job with the phospholipid bilayer, or in deeper sense, it works together with the fatty acid chain that form the hydrophobic tail. So, cholesterol function is almost the same with the fatty acid chain or the hydrophobic chain tail. Now we'll take a look on the function of the protein. Among all components, proteins carry out many functions for our cell membrane. First, they work as transport proteins. They also work as enzymatic activity, where they catalyze reaction within or along the membrane surface. They also work for signal transduction, in which they bind with hormones or other signaling molecules for information transfer. Apart from that, they are also important for cell-cell recognition, intracellular joining, and attachment to the cytoskeleton and extracellular matrix. Now we have covered the fluid mosaic model. Now we will cover the substance movement across the membrane. Now, we have the substance movement across the membrane are based on the selective permeability. And selective permeability is required by the cell for the food intake, for the excretion and secretion of the waste products, to maintain the ionic gradient, and to maintain the pH balance within the cell. First, we have the passive transport. Passive transport can be divided into three. We have the simple diffusion, osmosis, and facilitated diffusion. For simple diffusion, it is the movement of molecules down the concentration gradient. There are no energy required from the cell, and the passive transport will continue to occur until equilibrium is established or achieved. So factors that affect the diffusion rate are size, the shape, charge, temperature, steepness of concentration gradient, the distance, and the membrane surface area. So the animation here shows how the simple diffusion occurs. So the molecules that undergo simple diffusion are uncharged molecules such as oxygen or carbon dioxide, water, or liquid soluble molecules. For some molecules such as oxygen are non-polar, so they diffuses very quickly. For carbon dioxide, they are polar, but they are very small in size, so they can diffuse quickly. Water is also polar, but also very small, just like carbon dioxide, so they diffuses quickly. Next, we will take a look at osmosis. So for osmosis, it only involves the movement of water molecules down the concentration gradient, and it always involves a partially permeable membrane. The membrane is permeable to water, but not to solids. Otherwise, it only allows the passage of water. Here, aquaporin is involved as pathway for water molecules. So here, we have the hypotonic or isotonic condition. For hypotonic condition, we have more water but less solute. Solute here is the 2% supersolution. Therefore, to achieve balance between the two areas, the water will need to go inside to ensure that the water concentration is equal between both sides. For the second, the hypotonic condition is the condition where there are a lot of solute, 10% glucose solution. So the water will tend to go outside the cell to achieve equilibrium. Therefore, the balloon structure will be shriveled. But for isotonic condition, the water condition and the solid condition is in equilibrium. So the balloon shape is equal with the experiment. Now we will take a look for the isotonic, hypotonic, and hypertonic condition for animal cell and plant cell. While isotonic solution is preferred for animal cell, the plant cell will prefer the hypotonic solution because they need water for their rigidity and support. But for animal cell, they require isotonic solution because they do not have the cell membrane. More water will lead the pure red blood cell to be lysed, while hypertonic solution will lead it to be shriveled. So here is some of the animation for the hypotonic, isotonic, and hypertonic solution for both animal and plant cell. Here we have the another experiment for hypotonic and hypertonic solution. Remember, the membrane is only permeable to water, but not to solutes. Therefore, the water will move towards the area with high solute concentration to achieve the water equilibrium. That's why we see that the water level is not the same, but the water concentration gradient for both sides is the same. Next, we will go to the facilitated diffusion. Now, for facilitated diffusion, it always involves the lipid insoluble molecules, where these lipid insoluble molecules do not pass readily into the phospholipid bilayer. Therefore, these substances will pass through the membrane by a process known as facilitated diffusion, in which, for this one, the molecules will move down the concentration gradient, just like the simple diffusion. However, for facilitated diffusion, they require the help of transport protein. It can be two types, which is form or channel protein or carrier protein. Therefore, the diffusion for facilitated diffusion is highly rapid and highly specific. So here is one example for the facilitated diffusion. First, the glucose concentration outside the cell is higher than the inside of the cell. So the glucose will bind to the glucose transporter protein. The protein will change its shape and the glucose will be exposed to fluid on the other side of the membrane. It detaches and leaves the channel. So the glucose transporter protein will shuttle the glucose from outside to inside the cell to achieve the equilibrium down the concentration gradient. So here is the animation for the glucose movement. As you can see, the glucose transporter will change its shape accordingly to the glucose structure to allow its transport. So now we have covered the passive transport. Let us move to the active transport. For the active transport, it involves the transport of molecules across a membrane, but it is against the concentration gradient. When it is against a concentration gradient, it requires energy, and it involves the use of carrier protein. So here is the animation for the active transport. So first we have the calcium pump. For calcium pump, the protein will attempt to transport calcium from low concentration to high calcium concentration. In other words, it is against the concentration gradient. So first, the ATP will attach itself to the calcium pump. This will cause the ATP to transfer a phosphate group to the
to the calcium pump protein. Then this will allow the calcium to be shut